I thought of focusing on molecular base of inheritance and give special emphasis for transcription. For transcription, there is a PPT ready, but before that, I didn't want to start directly with transcription. Let's have a kind of from introduction till the end of the chapter. Coming to the introduction, in class 11 itself, Amuda ma'am and Gayatri ma'am, we should have insisted our children to draw the secondary structure. I hope we are doing it because in class 11 there is a chapter called biomolecules. So when we teach biomolecules chapter, we need to show them the structure of primary, secondary, and tertiary structure of proteins. We need to show them the structure of the nucleic acid. So I normally when I teach class 11 biomolecules, I make them draw the secondary structure, numbering the carbon atoms. I hope you are doing that numbering of carbon atoms, Gayatri ma'am and Amuda ma'am.
Yes, ma'am. Yes.
We need to number the carbon atoms so that they can understand the third carbon of the first sugar is connected to the fifth carbon of the next sugar through the phosphodiester linkage. So again, let us start the session with the importance of DNA, the nucleotides, the nucleosides, the complementarity existing between the nitrogenous bases, and insist that DNA is a polynucleotide. Tell the children what is called a nucleotide, why we can say DNA is a polymer of nucleotides. So DNA and RNA can be called as polynucleotides. So let them recall whatever we taught them in biomolecule chapter. So it is a polynucleotide made up of what is a nucleoside and what is a nucleotide. Nucleoside will be formed between the sugar and the nitrogenous base, and whereas the nucleotide will be formed between sugar nitrogenous base with the phosphate bond. And on the secondary structure of the DNA, let us recall, or let us again insist the children to mark the bonds. Where is the glycosidic bond? The glycosidic bond is found between the carbon atom, the sugar and the nitrogen base. And between two nitrogen bases, the hydrogen bond, double bond and triple bond are covalent bonds. And between two sugars, the phosphodiester bond. So let the children understand there are three different linkages. So in class 11 itself, we will teach all these in taking more time. So when they come to class 12, when they take the DNA concept, they should find it so easy. Because in class 11 itself, we have taught them the basics of drawing. We have taught them the numbering of the carbon atoms. We have taught them the bonds which is present in the DNA. The class 11, when we teach biomolecules itself, let us teach them the structure of the DNA molecule very clearly, making them understand the numbering. Let us number on the board. Number on the board the carbon atom starting from 1 to 5. 5 will be the fifth carbon will be outside the ring. Am I right, madam? Gayatri madam, Jensi madam, Amuda madam, hope I am correct. The fifth carbon will be outside the ring. So let them know the fifth carbon of the first sugar is connected to the third carbon of the succeeding sugars. So that concept should be taught very well in class 11. So let us also tell the children that the DNA what we are drawing is the most common DNA found in the living cells, which is the B DNA, B for boy. B DNA. So children will ask, why ma'am there is a B DNA? Let's make a note now. There are 5 types of DNA: A, B, C, D and Z. Are we making a note, teachers? There are 5 types of DNA: A, B, C, D and Z. What we are drawing is B DNA. And what is the basis of classification? The basis of classification of these different types of DNA is based on the number of base pairs.
Then turn, then
yes,
the helical turn.
Yeah, very good. Correct, Amuda. In one turn, how many base pairs are there? Based on that, we classify the DNA into A, B, C, D, and Z. The most common is the B form. And out of the 5, A, B, and C, D are right-handed helices, whereas Z alone is a left-handed helix. Am I right, Amuda ma'am?
Yes,
A, B, C, D are right-handed helices.
Only one alone is. Yes,
yeah, Z alone is a left-handed helix, and A has 11 base pairs per turn. B has 10 base pairs. C has 9 base pairs, and D has only 8 base pairs. Z has 12 base pairs. Should I repeat, teachers? A, B, C, D, and Z are the 5 types of DNA. What we are trying to teach them is the B DNA, which is very common in the living cells. And the classification of DNA is based on the, as Amudha ma'am also says, number of base pairs per turn and the nature of the turn, nature of the helix, whether it is a right-handed helix or a left-handed helix. A, B, C, D are the right-handed helices and Z is a left-handed helix. And A has 11 base pairs per turn, B has 10 what we are drawing, and C has 9, D has 8 and Z has only 12.
And is it alone has 12? Is it alone has 12 base pairs?
12, yes ma'am, 12 base pairs ma'am. Is it alone has, this is an additional information, Amudha ma'am. So children will be able to do
Yes,
neat
yes,
exam.
ma'am. Yes, I know that.
So let us not reach only from the reader. And let us see, all the children will be very familiar about Watson and Crick. But they may not be familiar about the previous scientist who also did lot of investigation on DNA, like Rosalind Franklin. Yes. And DNA as an acidic substance was identified by Miescher in pus cells. So Rosalind Franklin's, she was
Yes,
a
ma'am. Yes, ma'am.
lady scientist woman, and she did X-ray diffraction. And Edwin Chargaff. So what I suggest to my children is, I will advise them to take an A4 size paper and attach it in front of the molecular chapter, any chapter, biotechnology or molecular, any, before every chapter, I ask them to attach one plain paper. In that plain A4 size paper, I will make them write all the scientists' name. Like molecular basis chapter has many scientists' name, no madam. Like Miescher is there, Rosalind Franklin is there, Edwin Chargaff's constant ratio is there. The ratio between A to T and C to G is always constant. Edwin Chargaff, Watson and Crick, Frederick Griffith. Namaste, sir. Parthiban, sir. Welcome, sir. Parthiban, sir. Welcome. Frederick Griffith is there, Hershey and Chase experiment, Michelson and Stahl, Alec Jeffries, who discovered the DNA fingerprinting technique, and Sanger, who devised the technology of separation of biomolecules. All the scientists' names with the year, let them write on the plain sheet. This will help them during examination, during preparation. If they look at this plain sheet, they'll be able to recall all the important gist of the chapter. This I normally do for all the chapters, 16 chapters. They will attach one prefix, one white unruled paper. In the unruled paper, they will only write the gist. Then let us train our children to draw the single stranded polynucleotide, double stranded polynucleotide. But here there is a difference. Teachers in class 11, we would not have given much importance to show anti-parallelity. Whereas in class 12, we need to tell the children to draw both the nucleotide strands in opposite direction. They should draw one in 3 to 5 dash direction, the other in 5 to 3 dash direction. So if they know the numbering of the carbon atoms, they'll be able to comfortably draw the structure of the double-stranded polynucleotide strand. Then let us give more importance to the investigatory experiments to prove that DNA was the genetic material. Griffith's experiment. After Griffith's experiment, it's a very popular 3-mark question. The name of the scientist who biochemically verified DNA. McLeod, McCarthy and Avery. Am I right, sir? Parthiban, sir, it's a very frequently tested question. How did they biochemically verify Griffith's experiment? Using the enzymes. They used RNases, proteases, DNases, and only found that when DNases were used with the biomolecules which were taken from the S-strain bacteria, the transformation did not take place. So these McLeod, McCarthy, Avery, that also we can make them write on the plain paper. So before every chapter, let us advise the children to attach one plain paper, one paper cut to appropriate size of the reader, and write all the important informations. So that particular sheet, both the sides, 1 or 2 sheets, will give them the gist of the chapter. Before exam, they can only see these 2 pages and go. We'll tell the children to update name of the scientist, name of the discovery, year of discovery, importance of the experiment conducted by them. Like that, if you keep on making the, so if you just train them for 1 or 2 chapters, they will automatically start doing for the rest of the chapters. Then we have to give, all of us are doing, we are able to teach in a very effective manner. Hershey and Chase experiment, Michelson and Stahl experiment. But when we draw on the board, I hope all of us are using color chalks for Hershey and Chase experiment to show
Yes,
one
yes.
set of bacteriophages where the protein coat is radioactive, another set of bacteriophages where the DNA is radioactive. So let us try to use differentiating color chalks, and let us tell the children to carry crayons even for the coming exam. Anyway, this is not there for the for term 2 examination, but let us advise children to carry crayons. But the crayons should not belong to red or green color. Any other contrasting colors, let them carry for differentiating Hershey and Chase experiment. Similarly, Michelson and Stahl experiment, the heavy DNA, light DNA. I normally tell my children to use 2 colors. Some children, if they don't bring any color pencils to class, they will be advised to use pencil and pen. One DNA they will draw with pen, blue color pen. One the DNA they will draw with pencil. So let us, if you do it on the using color chalks on the board, children will automatically will take a color pencil. If we do everything with white chalks, then they will not get any idea to use a two distinguishing colors. So let us advise our children to carry 2 contrasting colors, pink and light blue, that light green and light pink. If they carry dark green alone, let them not carry. Some other brown and pink, if they carry, it will be useful for them to draw PBR 322, Michelson and Stahl experiment, Hershey and Chase experiment. In the examination, they will not lose mark. So let us, these are all simple experiments. So in Michelson and Stahl experiment also, again one scientist name is coming, Taylor and his co-workers. They verified the same concept in faba, Vicia faba, the beans. So the 1 mark questions are possible. Name the scientist to see, like how we train them for the biochemical verification for Griffith's experiment. We should also train them for the biochemical verification of Michelson and Stahl experiment by Taylor and his co-workers on Vicia faba. These are all informations which are to be underlined in the reader. Then coming to the molecular biological processes. So again and again, we have to insist the children to remember Crick's contribution. The Francis Crick's contribution of central dogma, how the DNA becomes RNA, how RNA becomes, so n plus 1 times we should say this in the class. Whenever possible, let us write the central dogma on the board. So the central dogma will never be forgotten by the children. They will always know DNA becomes DNA by replication, DNA becomes RNA by transcription, the RNA becomes protein by translation. So whenever possible, n number of times, let us repeat central dogma. Let us always write it on one corner of the board so that central dogma will never be forgotten. So they will know the flow of the genetic information, how the genetic information flows from DNA to RNA and through RNA to protein. And they will also understand why DNA depends on RNA for protein gene expression. And we'll always connect this to DNA expression is protein expression. So they will be easily understanding gene expression is nothing but protein expression, because DNA is nothing but a segment of gene. Or a gene is nothing but a segment of DNA. So DNA becomes protein through RNA. This way we can also make them understand how in the early organisms RNA was acting as a genetic material. Because at least DNA depends on RNA for expression. RNA does not depend on any molecule for expression. RNA can directly express into gene, express into protein. That is why RNA in the beginning was the genetic material. But there are certain properties of RNA which does not allow the RNA to act as a genetic material. So in this context, we need to make a table. Give the children the table of differences between RNA and DNA. Why RNA cannot act as a genetic material? Because of its labile nature. It can degrade very easily. It's very reactive because of its catalytic nature. It has got multiple roles to play. Whereas DNA can only act as a genetic material, but RNA can act as a messenger molecule, can act as a catalyst molecule, can provide in the ribosome molecule, tRNA can bring amino acids. So since it has got multiple roles to play, it cannot act as a genetic material. It can undergo mutation very fast. So a molecule which changes with time cannot act as a genetic material. So in this regard, we can make the children understand why is DNA the most stable genetic material. Because DNA does not undergo that quickly evolution. Still, it can undergo mutations, but the mutations are very slow. And in this regard, we can always say that is why the COVID pandemic, What we are experiencing now, COVID virus is a RNA virus. To find vaccine for such virus is a huge task, because by the time the vaccine is ready, the RNA would have, the genome would have got mutated. So that is the drawback of the RNA acting as a genetic material. That is why RNA evolved into DNA. So RNA, there is a paragraph called RNA world. So from central dogma, all these concepts can be taught. Central dogma, they can never forget any concept you relate to central dogma. So children will understand DNA expresses through RNA. Gene expression is protein expression. Behind every protein, there is a gene. And gene is nothing but a segment of DNA. Then when we teach the molecular biological processes, for example, we follow the central dogma, how DNA becomes DNA by replication, how DNA becomes RNA by transcription, how RNA becomes protein by translation. Author mentions 2 enzymes for replication, which are DNA-dependent DNA polymerase and ligase. But there are many more enzymes involved. I hope we are teaching all the enzymes, teachers, like topoisomerases, helicases, unwindases, editor enzymes. Editor enzymes will do the proofreading. Finally, the proofreading will be done. In spite of proofreading, if there are errors only, they will act as raw material for evolution. So if this chapter is taught well, evolution will be very easy, because during evolution we can make the children understand it is the variations which have happened in the during the DNA copying process, which are acting as a raw material and which are the source for variations. And variations accumulated over a period of time will lead to evolution. So while teaching replication, let us say that replication occurs in the S phase. So let the children recall the cell cycle. Replication occurs in the S phase, and some of its details are still not very clearly understood. So we know it is taking place in the DNA becoming double in the synthetic phase of the cell cycle in mitosis and meiosis, and only both the strands are copied, and throughout it cannot copy because it's quite long, 2.2 meters DNA. So packaging of DNA has to be recalled. The concept of nucleosome, chromatin, all that has to be recalled. Euchromatin, heterochromatin concepts have to be taught with a lot of importance, significance. So let us tell the children that entire DNA to open out becomes very energetically expensive. So part by part it opens, and replication begins in a region called origin of replication, ORI. And we will connect this to biotechnology. If this is taught well during molecular basis, during biotechnology teaching, we'll tell the children to recall why we are choosing a vector with ORI, because vector with ORI can only undergo replication. So if they understand replication now, while in the beginning of the year, they will understand biotechnology very comfortably. So selfishly, let us teach them very well in the molecular basis chapter, so that our job of making them understand biotechnology becomes little easy. That's why in class 10 itself, whenever I have a chance to handle class 10, I give them the basics of genetics very well, so that the children who take biology group, they will find genetics of class 12 quite simple. So whenever you get a chance to go to 9 and 10, make these concepts very strong, teachers. If in class 9 itself, teach them the cell, the basic unit, the cell chapter, the tissues chapter with all 11 standard inputs. So when the child comes to class 11, the child finds it very easy. Similarly, diversity of living organisms. In class 9 itself, let us teach them what is coelom, pseudocoelom, acoelom, coelom, all these concepts to be taught. If you get an opportunity to go to class 9 and 10, you please take it as an opportunity to give good foundation for 11 and 12, because most of the children get inspired in class 9 and 10 to take 11th standard. They see the biology of the class 9 and 10, and they are inspired to take class 11 in, they take class biology in class 11. Okay, so let us give the names of the additional processes, additional enzymes in this one, replication. And author is not explaining replication very clearly. Author says in one paragraph, he talks about the raw material. He says the time taken by that E. coli DNA is just 20 minutes. In 20 minutes, the entire DNA has to be copied. But let us teach them the procedure very carefully, right from unwinding. Right from unwindases, how the unwindases unwind the DNA, how the DNA stands separate, how the copying occurs, how DNA-dependent DNA polymerizes, and how the process goes on very effectively in one polarity and not effectively in the other polarity, how 5 to 3 is a continuous formation. 3 to 5 is not a continuous Okazaki fragments or discontinuous fragments. All that we will teach in detail. Let us take one complete period for application and 2 or more periods for transcription. Similarly, 2 or more periods for translation. These 3 are the molecular biological process where children will get doubt till the end of the year, till board exam. Though the board exam questions are going to be simple, they will find it very, especially the good ones, those who want to understand and learn will find it very difficult. So let us make it very comfortable for them. And coming to transcription, I have a PPT to show you. Before that, let us first tell them, unlike a replication where both the strands are getting copied, here in transcription, one of the strands is only copied. The 2 reasons why both the strands cannot be copied. Because if both strands are copied, both the strands are, so let us tell the children the difference between similarity and identicality and complementarity. Complementarity does not mean similarity. Both the strands are not identical. Both the strands are only complementary. So this the difference between identicality and the complementarity has to be given that input has to be given to the children. So we can tell them both the strands have different sequences. If they code, both of them are going to be copied, they are going to code for 2 different sets of amino acids. That will make the translation process very confusing. Whereas one of the strands is copied, one particular segment, one particular portion is copied, that portion is flanked, bordered by promoter and terminator sequences. So let us draw one highly coiled DNA on the board and let us tell them inside somewhere one region is to be copied. Let us show with color chalks. This region is bordered on one side by the promoter sequence, on the other side by the terminator sequence. So children will understand the entire process takes place within the nucleus, but the translation process takes place outside the nucleus. The mRNA molecule comes out. So whenever we teach replication and transcription and translation, I feel as a biology teacher, you should draw the cell on the board, entire structure of cell. Say draw one plasma membrane and draw the nucleus. So show them where these processes take place. So children will be able to conceptualize instead of simply teaching what is there in the reader. Let us take the child to the location. Let us take the class 12 child. So this is where replication takes place. This is where translation takes place. This is where transcription takes place. Because in board exam, sometimes they ask, where are the changes? Capping and tailing occur. Unless you, while teaching, if we don't insist, children will not know. They should know that capping and tailing also take place within the nucleus. Only after getting capped, only after tailed, it moves out of the. Why should it be capped? Why should the mRNA be capped? Because there are number of hydrolytic enzymes, enemies. They will dissolve the mRNA. So the mRNA has to come with a lot of protection. So what I normally say, because girls, I teach girls, I say, whenever you go out for a party, you really do a lot of makeover. Am I right? They do a lot of makeover, stand in front of mirror, look at their face number of times, they get their dress ready, get their accessories ready. Like that, mRNA will do nice makeover and come out. Am I right, teachers? So in this way, if you teach, they'll be able to relate. So with makeover, these changes are very analogous to making over, how the girls do makeover before they attend any function. The mRNA is going to come out for a very important translatory function for which it should be very, very protected. That is why capping is done at 5-N, tailing is done at 3-N. Then, before I go to the PPT, once teach them the differences between replication and transcription. And when we say the one more complication, if both the strands are copied, they are going to give rise to, or they are going to code for 2 different sequences of amino acids, and it is going to be 2 different proteins. Necessary amino acids will not be. May take an example, insulin as an example, salivary amylase is an example. If insulin has to be expressed, only insulin coding sequences have to come. So that is somewhere there in one particular portion of the genomic DNA. That portion has to be targeted. So, and one more thing, if both the RNAs are, both the strands are copied, they'll become complementary and they will become twisted, they'll become, they'll bind with each other. So, a double-stranded RNA cannot undergo translation. This நம்ம இங்க நல்லா சொல்லி கொடுத்துட்டா applications of biotechnology நம்ம வேலை ரொம்ப சிம்பிளா இருக்கும். Am I right, teachers? நம்ம சொல்லி தரோங்க. The pest resistance in tobacco plants, we are teaching them the novel strategy, which is nothing but mRNA getting silenced or RNA interference. And the concept inge solli kuduthurala number. எப்படி சொல்லி தரலாம்? If both the RNAs are becoming transcribed, they will become double stranded. A double stranded RNA molecule cannot undergo translation. இத நம்ம இங்க ஸ்ட்ராங்கா சொல்லி கொடுத்துட்டா அப்ளிகேஷன் சொல்லி தரும்போது we can simply touch upon, because children will already would have understood. Because dsRNA is silenced, that is why the particular protein is not expressed in the tobacco. That is why the tobacco plants become resistant. So, if in this way we are able to establish a connection between molecular biology and biotechnology. If molecular biology is well understood, biotechnology understanding will never be an issue. Again, I'm telling you teachers, molecular biology, biotechnology are the two units where the children find it little difficult before the board exam. And out of 2 chapters in biotechnology, chapter 1 is very hard for them, chapter 2 is comparatively easy. There also children will get confused in RNA interference. Am I right, Parthiban sir? N number of times children will be asking us.
Yes, ma'am.
I have experience, sir. What is your experience, Parthiban sir? RNA interference.
Yes, ma'am. They'll get confused only, ma'am. This one.
They will get confused.
Every time.
The best child has she was actually a very intelligent child. She was not at all convinced. Ma'am, how are you telling that it will occur, ma'am? So there, that is why when molecular basis solli tharumbodhu nama double stranded RNA pathi nama we can give nice input so that it won't undergo translation. Anga nama solli tharumbodhu this particular RNA, a very important RNA for the nematode or the protein we are able to identify. For that protein only it is coming to the roots of the tobacco, and the protein we are silencing, so the parasite is not able to survive. So please establish connections between molecular biology and biotechnology. Yes, teachers, I think I am.
Yes, ma'am. Sure,
Share your experiences, Jency madam, Gayatri madam. What is your experience in teaching RNA interference or transcription? What is
Yes,
the problem
ma'am.
that you have encountered? Yes, Jency ma'am.
Ma'am, whenever we are teaching this translation, if it is properly taught, means they understand RNA.
Yes, ma'am. Whenever we do transcription and translation properly, they will understand RNA interference very nicely. Amuda ma'am, Gayatri ma'am, please share your views.
Ma'am, as you insisted us to take the lesson last year, I have followed in the same way only, ma'am. Transcription and all repeated teaching, ma'am, again and again.
Yes, again and again.
Yes, ma'am.
Yes, madam.
Then our
Gayatri ma'am.
translation also like
Yes,
that.
ma'am.
only.
Amuda. Yes, ma'am. Amuda ma'am.
Even translation also in the same way you have told, ma'am. Same way.
Yeah,
I
in
have
translation also, author is not giving such a great explanation. We have to
Ma'am,
teach
I
them.
have one doubt, ma'am.
Yes, ma'am.
mRNA actually it is short-lived only, no? Only for 2 minutes it will be there, isn't it, ma'am? mRNA.
mRNA means actually, ma'am, you are asking about the gene. It is to be discussed along with gene expression, ma'am. It is a short-lived molecule, and depending on the physiological requirement only, it will undergo translation. If it has to be translated, it will come out of the nucleus through the nuclear pores, get attached by the ribosome, and start getting translated, madam. And that is also under control. The translation is also that we are teaching them. The gene expression can be controlled, regulated 4 different stages. One only we are explaining them. How the gene expression is controlled in the transcriptional stage alone we are explaining with the help of lac-operon. Other 3 are not explained for 12th standard level. They'll be learning in the graduate level. Yes, Amuda ma'am. Yes, Amuda ma'am. Any other, Gayatri ma'am, any other you experience that you encountered with your children? Because I have lot of issues with these chapters. Children will come back to me. In the class, they will be very quiet because they assume they understood everything. When you announce a test, they will start learning.
Yes, ma'am. Same thing, ma'am. When we explain the topic transcription translation, we used to explain step by step. That time they will be like it has been understood everything.
Yes,
But the same thing what you told when we ask them to write a test, when they are learning together some particular chapter for revision exams, they are forgetting these concepts and they are finding this concept difficult one.
Very difficult. And molecular basis quite long, ma'am. Very long chapter. So adukku enna pannalam, ma'am? What I suggest?
No, because terms are very new to them. First of all, so far they would have been learned as cell, nucleus, inside nucleus, chromosome. That's it. Now we are teaching in depth what's happening inside chromosome. We are teaching them in depth, and all the terms are very new to them. Cell and all they are learning right from class 6 onwards. They are learning, but this is all new terms. What they are learning.
Totally new, totally.
Yes,
That's
totally
why I
new.
think in biomolecule chapter, we importance 11th standard we will teach about DNA in a much detailed manner. So in 11th
Yes,
itself, we will make them understand the salient features of DNA. And as you say, when you give a test in the whole molecular basis chapter, they are going to make a mess only, madam. So what I suggest is give them slip tests. Replication mudichone or replication kuduthudlam. So you give them some hypothetical DNA, ask them to make one strand. You ask them which is the template strand, draw the coding strand, all that you before that. Replication, you give them the complement, you give one sequence, let them do let them understand complementarity. So give them lot of hypothetical examples on the board to keep on, let the child take a piece of chalk, you give one portion of DNA, let them do the other portion of DNA, let them understand the Erwin Chargaff's ratio. The ratio between A to T and G to C is constant. We can say no, a particular DNA has 20% of adenine. Or chinna kutti problem. 20% of adenine. Calculate the percentage of the other nitrogen bases. So when they know 20% is adenine, they will definitely know 20 other percent will be thymine. So 20 plus 20, 40 is over. Remaining 60, 30 plus 30. So simple sums. So after replication is over, conduct a test in replication and let them draw the replication fork. Replication fork with labeling. So the newly synthesized strands, the discontinuous strands. What is the use of ligase? Ligase is a molecular gum. Like restriction endonucleases are molecular scissors. Let the children understand it acts as a glue. It will only attach the discontinuous fragments. So give a test after replication. Give a test after once you teach transcription, let them draw the transcription unit with polarity in the next class. Class work notebook le panna solluvom. Even if the child sees the previous class work, it's okay. Let the child have the confidence of drawing the transcription unit, marking promoter sequence, terminator sequence, template stand polarity, coding stand polarity. Why template is called a template stand? Why coding is not coding? Why it is displayed? So let the child will understand the mRNA formed will be exactly like the coding strand, but for the presence of U in the place of T. So when we teach them RNA DNA difference, let us teach them about the uracil. So children should understand the newly formed mRNA molecule will be exactly like the coding strand. So let's give lot of exercises. You give one template strand, make them write the mRNA in the proper polarity. Give them the coding strand, make them write the mRNA. Give them the mRNA, make them write the coding strand. Give them the mRNA, make them write the template strand. So don't give them the typical questions like draw only the coding strand. You give them some coding strand. Ask them mRNA. You give them mRNA. Ask them for the template strand. So make their mind work. Let them understand mRNA will also have the same polarity 5' to 3'. Because it is from the template strand 3' to 5, 3' to 5'. So you give U, wherever U is there, they should write T in the coding strand. So make the only our quality time has to be spent. Let us not rush through at all. And similarly, when you teach translation, again the book author is not very great, giving a great explanation. We have to talk them about the process, the machinery, the enzymes, the procedure. Even for replication, the definition of the process, machinery, what are the raw materials involved, how the raw materials are used, and children will ask, where are the raw materials coming from? One and only source is food. You just connect it to digestion process. When we eat food, all the nucleic acids are broken down into the nucleotides. These nucleotides are there in the nucleus. Even now, whenever they are working out a derivation, they are working out a mathematical problem. One new mRNA is formed. Because for understanding, so like that, no, that's why I tell you, till we finish translation, before we start teaching the class, there must be a cell diagram on the board. There must be central dogma. So that you ask children, what are we learning now? Where is it taking place?
Yes, ma'am. Even I used to ask the same question. First of all, we should make them clear why we are learning this. Why every chapter, before every chapter, we should create some interest why they should learn. Otherwise, they will think it as something like a new topic, like how in other subjects they are learning. They would not think like that.
They will not connect it to that. You see, even now some new transmitter. See, whenever they want to understand concept, a new neurotransmitter is made. Neurotransmitter is nothing but a protein. That protein only is coming from the mRNA. Like that, you keep on rating. So till you finish the chapter, let us draw one cell diagram on the board. Let us write central dogma. Let the children know this particular process takes place in this particular part of the cell. And this when we teach gene expression regulation, tell them what all levels at which the gene expression can be regulated. What is called gene expression? Gene expression is protein expression. Even that can be understood only by the central dogma. Molecular biology, because in the before the OTT online teaching training sessions, some schools requested some topics like transcription, so I made only for transcription. Anyway, we are going to discuss the whole chapter. So definition of transcription, please go through. Cellular process in which RNA is synthesized using DNA as a template. The process called transcription. So it shows the entire central dogma, how DNA becomes DNA by replication, how DNA becomes RNA by transcription, how RNA becomes, say, all these are amino acid sequences. Then the next slide. Ribonucleic acid, a polymer of ribonucleotide held together by 3 to 5 phosphodiester bridge and are single stranded. Let them again and again let us insist the fact that RNA is a single stranded molecule, DNA is a double stranded molecule. This molecule is known for its storage and transmission of genetic information in catalysis. It can act as a multiple roles of RNA, catalyst molecule, messenger molecule, genetic material, all that can be insisted. All RNAs, what do I mean by all RNAs? mRNA, tRNA, ribosomal RNA. Except, it should be except RNA genomes of certain viruses derived from information which is stored permanently in DNA. So the information in RNA is stored in DNA. These are the different types of RNAs. mRNA 5 to 10% transfers the information of gene to ribosome. It is a template for protein synthesis. We can always say mRNA is a template for protein synthesis. tRNA, it reads the code on mRNA and transfers the appropriate amino acid to mRNA, AA for amino acid, and rRNA constitute most of the ribosomes. Many additional specialized RNAs which have catalytic activity or regulatory functions are present in the cell. Replication versus transcription. We can make a table in the children's classwork notebooks. What happens during replication? Entire DNA molecule is copied, taking place during S phase of the cell cycle. Whereas in transcription, a particular gene or the group of genes copied at any time, and some portions of DNA are never transcribed, non-coding sequences. Gene is a segment of DNA that codes for a type of protein or for RNA and may be present on any strand of DNA. So, 1 second. So let us locate the DNA, it's gene on DNA using color chalks. Differences between replication and transcription. Teachers, primers are attached during
definition of primer in easiest understanding way.
Yes, ma, I'll tell you, ma. See, how we, 1 second, let me minimize it, let me come back. I'll come to the presentation a little later. Yeah. Primers are oligonucleotide. They are also nucleotide sequences.
The definition, like chemically synthesized oligonucleotides.
Yeah, what I teach them, no. I will ask them a question. Have you observed a painter painting? All will say yes. Before the paint is applied, they will apply primer. Am I right, sir? Am I right, teachers? Before the paint is applied, primer podrom wall.
Okay.
Primer podrom. The primer pota paint nalla otikkum. Job of the primer is to make the nucleotides attach properly. Primers will bind. First primer will bind so that the next will Next, our nucleotide will bind. Then once the nucleotide binds properly, primer will get out. So these are not explained by the author. So you teach like this, teachers. Primer, or see, whenever we do painting on of the wall, or first we apply primer. Primer job is to make the paint stick to the wall or the door surface properly. Adhe madri, primer in molecular biological process are nucleotides which will first bind. Their binding will make the actual nucleotides bind properly. This is enough for a 12th standard site. Is Gayatri ma'am?
Yes, ma'am.
Shall I go back to the?
So what is the nature? What is the nature of that?
They are nucleotides and they will also have complementarity, ma'am. They are synthesized temporarily in the nucleoplasm. They are freshly synthesized nucleotides. Just see, that is why when you teach
so
machinery,
that should be complementary to the template and primer should be complementary.
Otherwise it will not be able to bind. Otherwise it won't be able to bind. So
okay, ma'am. And to that primer complementarity only new strand will be created, ma'am.
Yes, ma'am. So primer will first bind, followed by the nucleotide. Primer binding will allow the nucleotide binding very comfortably. And they are all present in that is why the cell will prepare itself. That is why S phase will take place. That is why the interface takes such a long time. Once the interface is done, when mitosis takes place dramatically, because all these are time consuming. Almost in 124-hour cell cycle in typical mammalian cell, 23 hours will be taken only for the interface. Why? Because all these have to take place. The raw materials have to get ready. That is why I teach replication with all the subheadings. What is the definition? What is the machinery? What are the raw materials? How the process goes on? What is happening at the end? At the end, so many wrong bases will be there. So there is an enzyme called editor enzyme. It's like how we do proofreading. You tell the children, they're all acting as captains, vice captains. They'll be doing some school magazine work. For school magazine work, there is an editing team. There is an editing team there. These editor enzymes will go through whether properly replication is carried out or not. Then wherever there are wrong bases, they will remove. Then they'll put the correct base. All in spite of all these things, if there is a wrong base, that is only called variation. So the editor enzymes will try to remove all the wrong bases and put the correct bases. But in spite of the editor enzymes efficiency, because the problem is again every molecular biological process is time constrained. I'm given 1.5 hour session today. I won't be able to do molecular base of biology because I'm time constrained. Because it needs lot of time. We need to have so many sittings like this to understand how we can effectively teach. So, because there is a time constraint, the editor enzymes also do the work very fast. And when they do, for this also I do an activity, Gayatri ma'am. I write one complex spelling on the board. One biological name, Propionibacterium sharpen, some complex name I write. Okay, first I write on the board. I tell the children to see it for a second. Then I rub the board. Then I tell the children to write. How many of them will write carefully, correctly? There will be some children who are writing correctly. Like this, there are sometimes replication taking place correctly. Many children would have written a E for A or I for yes or no, because I am given a complex word on the board. I do this in the class. So like how you are making me, because I am giving you only 1 second. And the 1 second la the board paatha correct, the குழந்தைங்க எத்தனை பேர் கரெக்ட்டா எழுதுவாங்க, நிறைய பேர் எழுதுவாங்க, சில பேர் எழுத மாட்டாங்க. That is how it takes place. These are all some tips to make the concept reach the children. You try this year. You try doing this year. You'll be able to, yes, see, every year we follow some different strategy to make the concept reach the child. Am I right, Gayatri ma'am? Shall I proceed?
Yes.
Shall I go back to the presentation? Yes. Primers are used for replication. Primers are not used for transcription. DNA dependent DNA polymerase, DNA dependent RNA polymerase, product, base pair, proofreading. See, I told you no proofreading happenings. Proofreading is done by editor enzymes. Shall I?
Why primers is not used for transcription? The purpose of primer is like
yes, ma'am. Yes, ma'am. Why? Because see, in transcription, the promoter sequence is there, no, ma?
Yes.
The promoter sequence provides the place for the RNA polymerase to bind. Once RNA polymerase is identified, because here replication, as you know, the entire DNA has to be copied. Entire DNA, both template strand and coding strand, entire double stranded DNA has to be copied. Here only a particular portion has to be copied. That particular portion coding for that particular protein has to be copied. So for which the promoter sequence will be helping the RNA polymerase to bind, then automatically it starts the polymerization. But here it is, primers are required for continuous binding of the nucleotides.
Okay.
Shall I go to the next slide, teachers?
Yes.
Features of transcription, highly selective. In the sense, only a particular region in the DNA is copied. Selectivity is due to signals embedded in the nucleotide sequence of DNA. The entire 2.2 meters DNA need not be copied. One particular small stretch is copied. Specific DNA sequence marks the beginning and end of C. Specific sequences marking the beginning and ending. Am I right, teachers? Here, that is, they are promoter sequences, terminator sequences. Signals instruct the enzyme where to start and stop transcription, when to start and how often to start. All this C. This third point, when the translation, when the transcription should start, depends on the physiological dynamicity of the cell or the requirement of the cell. Many of the RNA transcripts are synthesized precursors, primary transcripts which will not undergo maturation. They will have exons and introns. Transcription taking place in prokaryotes is cytosol. There is no cytoplasm and nucleus there. Everything is called cytosol. Whereas in eukaryotes, nucleus and mitochondria. Mitochondria also it takes place. Chloroplast also it takes place. It's not mentioned here. So I can enable writing. I can write here chloroplast also. In the nucleolus, rRNA is synthesized. In the nucleoplasm, tRNA and mRNA are transcribed. It's only transcription. Shall I go to the next slide, teachers?
Okay, ma'am.
Yes.
Procedure.
Yes, ma'am.
Yes. The basic biochemistry of RNA synthesis is similar in prokaryotes and eukaryotes, but its regulation is more complex in eukaryotes. And probably because of the complexity of the body design, prokaryotes have a very simple cell structure where there is no post-transcriptional stages required. Whereas RNA synthesis in prokaryotes is catalyzed by single large enzyme called DNA-dependent RNA polymerase, which will do initiation, elongation, and termination. But here it is done by 3 different enzymes, 1, 2, and 3. And another difference features in prokaryotes, polycistronic DNA, mRNA, here monocistronic mRNA. 1 second, I will stop sharing. I'll come back to it later, little later. Yeah. We teach structure of DNA in class 12. In molecular basis chapter, teach them the concept of cistron. So how do we introduce the concept? Because again, this is the area where the children will be really confused. So you start giving different definitions of DNA, gene. How gene was described by Mendel. Mendel called gene as factor. Am I right?
Factor.
Because in the previous chapter, you have taught them principles of inheritance. How Mendel called gene as factor. Even today, it's right. All the definitions are right. How did we define the gene in the beginning of the chapter? We defined DNA as the small segment, gene as the small segment of DNA. It's also right. Gene can be defined as a portion of DNA encoding a protein, correct? Gene is nothing but a portion of DNA but governing the protein, because again you connect it to central dogma, gene expression, protein expression. Behind every protein, then see, we have got 30,000 to 40,000 traits. We are very complex organisms. We are typical eukaryotic complex. We have a very intricate body design. Our body has n number of traits, and behind every trait there is a protein. Behind every protein there is a gene. So genes code for the protein definition is also right, but the modern concept says gene is a cistron. A cistron can be a polycistron or a monocistron. What is a polycistron? One long stretch of gene coding for many proteins. இதை இங்க நல்லா டீச் பண்ணிட்டா லேக் ஆப்பரான் டீச் பண்ணும்போது ஈஸியா இருக்கும் நமக்கு. பிகாஸ் லேக் ஆப்பரான்ல என்ன சொல்லுவோம் நம்ம, Z, Y and AMO. Z will code for beta galactosidase. Y will code for permease, A will code for transacetylase. So one long mRNA coding for many proteins. Polycystron. Poly means many, whereas why polycystron? They have a very simple body organization. They are only made up of a cell, prokaryotes. They don't even undergo transcription fully. By the time the transcription is over, translation is started. They don't undergo capping. They don't undergo tailing. Everything happens in the cytosol. So you teach them monocystronic concept is there in eukaryote. There's a clear-cut division of labor. One gene, one protein. Polycystronic condition is there in prokaryotes because of the simple body organization, cellular organization. One long mRNA coding for many proteins. இதை இங்க நல்லா சொல்லி கொடுத்தா லேக் ஆப்ரான் சொல்லும்போது ஈஸியா இருக்கும் நமக்கு. SRO teachers, when we teach lac operon, the entire DNA, mRNA, the same promoter sequence. And the sketch பார்த்து பசங்க பயந்துப்பாங்க. அந்த lac operon போடும்போது அவங்களுக்கு ask them to go back to transcription unit. அதேதான் அது. ஆனா additionally there are some regulatory sequences near the promoter. These regulatory sequences can either act as inhibitors or act as positive. They can either accelerate. They can be some. There you can also teach them. I does not stand for inducer. I stand for inhibitor. Always there is an inhibitor protein. That is why it is the lac operon is always in the switch off mode. So you develop a kind of connectivity link between the polycystron here and the lac operon. Polycystron in the குழந்தை நல்லா புரிஞ்சாச்சுன்னா lac operon புரிஞ்சிக்கிறது ரொம்ப easy. Because lac operon is a polycystron. One long mRNA coding for beta-galactosidase, transacetylase, permease. So, in this particular paragraph which is coming after transcription unit, let us give them all definitions of gene, right from factor till cystron. And cystron has got two components, exon and intron. What are exons? Exons are EX for expressed ones. Exons are there in the matured gene, whereas introns are interrupting sequence. I and inflex, they are not needed during processing. That is why introns are removed. So a cystron will have exon and intron. That is why the particular mRNA found in the nucleus is called HN-RNA, heterogeneous nuclear RNA. Heterogeneity. What is heterogeneity? It is not homogeneous. It has got unwanted sequences. Unwanted sequences are introns, which have to be spliced out, which have to be removed. So when you teach cystron, all these you need to teach so that children will be understanding it properly. Only thing is we need to take time and energy and effort to make the lesson little easy for them, because most of the places author is not very explanatory. He is clear, but he is not explanatory. So teach them the concept of different definitions of gene. You give them right from Mendel's factors till cystron. Classify and tell them cystron can be homogeneous. It is a homogeneous, heterogeneous. Homogeneous will have only exons. Heterogeneous will have exons and introns. That is why the mRNA precursor mRNA is called heterogeneous nucleus. Why heterogeneous? Exon, intron irukku. ஒரு வார்த்தையை break பண்ணனும். போர்ட்ல எழுதுறச்சே you break. Heterogeneous because exons and introns. N stands for nuclear. Still it is in the nucleus, children. It has not gone out of the nucleus. It has to undergo some processes. What processes? Capping, tailing. It has to get rid of the introns. Splicing. It will become homogeneous. Till it is not done, it is heterogeneous. The still introns are removed, not removed. It is heterogeneous. So H for heterogeneous, N for nuclear. It is still in the nucleus. Once it has to go out of the nucleus, it has to get spliced, it has to get capped, it has to get tailed. Capping is always done at 5' end. Tailing is always done. And the 5'3' end வந்து நம்ம DNA சொல்லி கொடுக்கும்போது clear-ஆ சொல்லி கொடுத்துடணும். And the numbering of the carbon atoms. 5th carbon is outside the ring. Up to 4, the carbon atoms are inside. 5th is outside. So how the 5th carbon is connected to the 3rd carbon? And the concept clear achina, children will not undergo any mistake in polarity. And whenever they draw a DNA strand without polarity, do not give them mark till revision examinations. Pre-board வரைக்கும் 0 மார்க்கே கொடுங்க. அப்பதான் குழந்தை கரெக்டா பண்ணும். முதல்ல நம்ம ஒரு அரை மார்க் கிரேஸ் மார்க். Once we start giving grace marks, children will not know to correct the mistakes. Polarity illana 0 mark you award. Be very strict in test corrections. Be very strict in quarterly, half yearly revision corrections. Let the child understand polarity is very important. Without polarity, if I draw DNA, my teacher is not going to give me mark. Be very strict. Only then we will insist the children indirectly to go to reader number of times. RNA polymerases use one of the DNA strand as template, which has a polarity 3 to 5, on which the complementary ribonucleotides are incorporated to synthesize RNA. The strand of DNA which is transcribed to RNA is called template strand. Opposite strand is called coding strand. When we teach coding, see this, there is a diagram here. Coding strand and the mRNA are very similar, but for the presence of D in the place of U. Inga U irundha anga T irukum. Otherwise, mRNA formed is exactly like the coding strand. So by giving number of exercises, every day 5 minutes you give them exercises. You give on strand, ask them to do a replication for it. Ask them to write a complementary strand. You give them 3 to 5, let them do 5 to 3. You give them hypothetical DNA strands. Number of times let us practice. And all the, why coding strand is very important. All the positions are defined with respect to coding strand. Promoter is at 3' end. Terminator is at 5' end. The solo number. Because every position is defined with respect to the coding strand. Next one. Please go through that. Should I zoom it, sir? Is it okay? Can you all read?
Yeah, we can zoom, ma'am.
These are all some additional informations, but still I prepared because no, we can. If we know more than what is there in the reader, we will be able to do a good job. Always I feel. So let us learn. Let us keep ourselves updated, teachers. Let us learn more. Let us go through more information about RNA polymerase.
If possible, please share it in our group also, ma'am.
Definitely, sir. I will share this in the group, sir. Because it will be helpful for us to teach. Thank you, sir. I will definitely do it. A single RNA polymerase performs multiple functions in transcription, such as bind to promoter site, unwinds a short stretch of double helicals. Unwinding is very energetically expensive. Select correct ribonucleotide and catalyze the formation of phosphodiester bond. Detects termination signals. Interacts with activator and repressor proteins. So if you teach here, they'll understand lac operon better. Transcription in prokaryotes. There are 3 stages: initiation stage, elongation stage, termination stage. It is dependent on sigma factor and rho factor. RNA polymerase recognizes the promoter site. Elongation phase, RNA strand is continuously growing. Termination, the RNA polymerase stops synthesis. And nascent means at the time of birth. Just born, newly born RNA. Initiation involves interaction of RNA polymerase and DNA at specific site. Sequence of DNA is needed for RNA polymerase to bind. So that means the RNA polymerase needs to search for the promoter sequence. The promoter has a characteristic sequence of DNA that directs RNA polymerase to initiate the transcription. It is usually located on coding strand. The simplest form are found in prokaryotes. Two general types of sequences elements are found. One sequence element is believed to promote the initial binding of the enzyme RNAP for RNA polymerase. Other element, these are all little high for the 12th standard children. Let us not show them. Let us only keep it for ourselves. All this will confuse the children. It's only for us. Initiation, sigma factor. The sigma factor will temporarily bind. The binding of the sigma factor is transient, or the sigma factor helps the RNA polymerase to detect the promoter sequence. Similarly, the rho factor will help the RNA polymerase to come out at the termination site. I will edit this and send Parthiban sir. I will do some editing because I did it in a hurry today. I'll do some editing and send it in the group. Am I okay?
Okay.
Is it okay, sir? Yeah.
Okay, okay. No problem.
Elongation, nascent RNA, 5-methyl guanine triphosphate, it is eukaryotes, 5-GPP. But we teach them only row dependent termination. All these are only for us. Let us not tell the children about the two types. Unless the child goes for NEET classes, it is not required. Row dependent, row independent. We only teach them the row dependent termination. So row factor helps in termination. Always use color chalks for the coding strand and the template strand. Use a color chalk for the mRNA, nascent mRNA. This is row independent, not needed for the children. Transcription eukaryotes, very important for a 3-mark question or a 5-mark question. The 3 types of RNAs. RNA polymerase number 1, RNA polymerase Roman number 2, RNA polymerase. Let us teach the children the expanded form of mRNA, hnRNA, small nuclear ribosomal RNA, transfer RNA. All 3 are very necessary for protein synthesis. Transcription eukaryotes is very complex. There is a clear-cut division of labor. There are post-transcriptional processes, capping, splicing, and tailing. Post-transcriptional modifications. The nascent RNA, also known as primary transcript, needs to be modified to become functional RNAs. Primary transcripts are heterogeneous because they contain exon and intron. The cistron concept. hnRNA are larger than, because matured RNA, it may be tested in 1-mark question. Why are hnRNA larger? Because they contain also introns, whereas in the mature mRNA, introns are not there. Mature mRNA is shorter. Splicing. Addition of 5-methyl guanine triphosphate, 5-MGPP நம்ம சொல்லி தரணும். 5M, 5 for 5 M for methyl guanine triphosphate, polyadenylation tail or polyadenylate residues, RNA editing. What I do normally for splicing, I think you all see one black color thread with me? Yeah.
Yes, ma'am.
What I do, I take any colored thread, sir. Either like this or a white thread also I take. I just mark some portions with either color chalks or ink. I make some portions like this. Can you see the white portion here? I'll make some more white portions. I'm using a black color thread here and white chalk I have at home. I am just trying to show you the heterogeneous RNA, heterogeneous mRNA. This will I do in the class also. One second, you please wait. I'm making a small aid. Suddenly I thought of it. Yeah. Now can you see the white and black colors here? White and black. Can you see the white stretches?
Yes, ma'am.
Yeah, like this I do in the class. I take a white thread. Using color pens, I shade some portions. So I tell the children, these are the introns. The white, in my example today, the white portions are the introns. The black ones are the exons. So you can demonstrate with the help of a thread. Then what I do, I loop it. See, make one loop. Make one loop. Wherever the white portions are there, I make a loop. Can you see 2 loops? Can you see 2 loops?
Yes, ma'am.
Then you pinch it. இந்த மாதிரி தான் நடக்கும் ஆக்சுவலா. mRNA என்ன ஆகும்னா mRNA will contain introns. The introns are twisted, means they will be spliced. What is splicing? A small loop will be formed and the loop will be pinched off. So you in the class you can show them one thread, and in the thread you can make some colored patches, contrasting colored patches. Those are the introns, and you show them the loops. Show them the loopings. Like this, it will be, it's called tail, it's called splicing. Splicing can be demonstrated. So just pinch it off. Then that will be removed. So what will happen? All the introns are away. Only the exons are there. After looping, it will be only black in color. It is homogeneous. Before it was hetero. So children will understand the heterogeneous nuclear RNA concept. These are all simple aids. And in this regard, I also want to tell you, when I teach meiosis, phase 1, which is very complicated, I take threads. On the previous day itself, I make white threads with some blue ink, or I dip the threads in blue ink or red ink. So I show them homologous chromosomes. One normal, I take through 2 threads and make one knot. So they are the chromatids. Centromere I can show with the knot. The knotted portion is centromere. So I have white chromosome, I have a red chromosome. Red is either red or blue or any other thread. So I can show them homologous chromosomes. They come together. I just call children in 4 or 5 numbers because there are many children. I can't show. So in small groups I call them. I draw on, I keep them on the table. And on the table I show the crisscrossing. One white thread, how the white thread intertwines with the red thread to form the chiasmata. These are certain very simple aids. Children will understand how crossing over takes place. What is the meaning of homologous chromosomes? How we can show simply using, see, these are all not very costly aids. Taking a thread is never, always we have examination threads. Same examination threads can be used for chromosomes, mRNA. I normally show them mRNA splicing. Splicing is a loop will be formed. That is why the author shows round compare. If you have a reader, you can check. During splicing, there are some spherical structures drawn. They are all nothing but the loops. The loop will be pinched. So all these are post-transcriptional processes. How a cap is given at a 5' end, how a tail is given at a 3' end, how the heterogeneous mRNA becomes homogeneous, then it becomes ready for translation. Then it comes out of the, that's why I tell you, please draw one diagram of the cell and the nucleus, show them the nuclear pores. Through the nuclear pore, mRNA comes out. Very close endoplasmic reticulum is there, because on the nuclear membrane, endoplasmic reticulum is there. The rough endoplasmic reticulum has the ribosome. Tell them that is why children, the rough ER is in close association with the nucleus, because it has to take part in translation. So if you relate like this on the board with cell diagrams, children will never forget the basics. If you have any other methodology, you can also share with me how you teach this concept in class. Any other aid if you are using, please share. Let us discuss. This is what I do in my class. I generally use diagrams using color chalks. I use a color chalk for one color for coding strand, one color for template strand, one color for mRNA, one color for DNA dependent, that the row factor. I always carry a box which contains white chalks and the other color chalks. I don't send my children. I carry my chalk box is very unique to me, so I will always carry a color chalk box, so that all these are taught well using the color. So children will also, many children will follow us by drawing color diagrams. So this is how transcription is done. So transcription in prokaryotes, transcription in eukaryotes. Then you stop for some time, then introduce them to genetic code. For so far, replication and transcription, they were easily understanding because there was something called complementarity. One nucleic acid was copied to another nucleic acid. Either one nucleic acid was becoming the same nucleic acid, either DNA became DNA or DNA became RNA, because there was complementarity. Now, the nucleic acid sequences are going to become amino acid sequences. Absolutely no relationship. How will they understand? That is why making them understand the importance of genetic code. Genetic code was proposed to bring out the relationship between the sequence of nucleotides on the nucleic acid to the sequence of amino acids on the protein. How a polynucleotide becomes a polypeptide. Teach them the role of all the scientists. Cell-free system, Nirenberg, Every band, it is advisable to give them the expanded form of all the 20 amino acids. I do it in the class. There is some space, working space near the genetic code checkerboard. I teach them to do the checkerboard also. Simple it is. UGAC, they can take three nucleotides. They can arrive at the entire genetic code. I hope you are also doing it, teachers. Let them decipher it. Let them not memorize, but let them know how the genetic code was proposed, how it was a combination of 3 nucleotides, how it was a triplet, why genetic code is unambiguous, why genetic code is degenerate. Give them examples. Tell them the expanded form of all the 20 amino acids. Not very difficult. Not difficult. We can say them. LYN, LYS lysine, isoleucine.
Biomolecules chapter itself, I used to tell them, biomolecules chapter while teaching the protein structure. That time itself, I used to tell them this amino acid name and all. That will be fine.
20 amino acids. Yes, Gayatri madam. In 11th standard, if your job is 12th, anyway, they make the child write the amino acids in 12th standard book. In our person 11th standard book, என்ன பண்ணுவோம் அந்த 11th standard information அந்த genetic code பக்கத்துல எழுத வச்சிரும். All the 20 amino acids அத வச்சு make them under what is called degenerate, what is called unambiguity, what is called universal nature. So make them under genetic code is very interesting part. Children will not find it difficult. So the salient features of genetic code with examples. UUU always codes for phenylalanine. It is same from bacteria till man. So it is, the code is universal. The dual function of the codon AUG. Children will generally ask a question. My children will ask, ma'am, will all proteins will have a methionine in the first place? They will ask or not? They will ask. Because methionine is the initiated codon. So the concept is every translation will begin with methionine only. Adla doubt kadayadma. Anal at the end of the translation process, how the RNA molecules are undergoing some maturation, how the DNA molecules are undergoing some feeding. At the end of the polypeptide formation, depending on the necessity, the methionine will be kept or removed. This concept is not given by the author. Author will only say dual codon solvar avladhaan, but children will get it out. They will think all polypeptides will contain methionine. Not necessary. For the process, it is necessary. For translation, it is necessary. For the mature protein, need not have methionine in the first place. And again, you recall 11th standard information. First end is called the N end. Last end is called the C end. Yes, sir. N end of the N terminus, C terminus. அதை நம்ம இப்ப நல்லா சொல்லி கொடுத்துட்டா, when they draw antibody molecule, see how every lesson is connected. Antibody molecule வரைவாங்க, அதே மாதிரி இன்சுலின் molecule வரைவாங்க. நம்ம biotechnology we will draw insulin molecule, A chain, T chain, C chain, C chain is மூடுன்னு சொல்லுவோம். இங்க antibody molecule வரையறச்ச there is N end and C end இருக்கு. புக்ல பார்த்தா கேர்ஃபுல்லா. பட் நம்ம சம்டைம்ஸ் நம்மளே அவசரத்துல பார்க்க மாட்டோம். பட் இந்த எக்ஸாம், இந்த கிளாஸ் டெஸ்ட் குழந்தை antibody molecule வரையறச்ச N end C end இல்லனா மார்க் கொடுக்காதீங்க. Let them understand antibody molecule is a polypeptide. Like any polypeptide, it will have a first end amino acid and the last amino acid. In the மாதிரி conceptual strength oda molecular basis teach panna, child will understand antibody molecule properly, later insulin molecule also properly. Every lesson is connected. So how we can teach them? Every translation will begin with methionine, children. But at the end of the translation process, the protein will be undergoing some changes. Polypeptide. And it will be taking place in the Golgi body. Golgi body will undergo, the molecules are modified. And the time, methionine venuna, methionine will be retained. Methionine vendana, methionine will be removed. So all polypeptides, all proteins did not have methionine. But methionine is very important for starting the process. These are all some hidden information which are not there in the book. Adanalda, this book, the lesson looks very difficult. There are many places where the information are not explicitly told by the author. Nammaloda job na adu. We should only bring the connectivity. So genetic code, now you take one full period to make them decipher the genetic code. Like how we teach the right hybrid cross, make them understand how the UAA comes, UAC comes, first position, second position, third position. Let the children not memorize, but they should know 5 or 6 important codons. They should know AUG, they should know UUU, they should know the 3 stop codons. UAG, UGA, and UGG. These are the stop codons. Stop codons, definitely they should know, because in the examination they get questions like stop codons. So they should memorize only the 3 stop codons, AUG codon, and tRNA molecule, SCR, serine codon is there. Few codons alone, let them know. Let them not know the code, 64 codons. And from that, they will understand the 3 stop codons, there are only 61. So more codons than required. 61 codons code for amino acids, 3 don't code for amino acids. Then tRNA molecule. Tell them tRNA molecules are like our specific transport means. How my auto rickshaw takes me from my place and puts me in the school. That they will take the amino acids from the cytoplasm. The child will ask, where are the amino acids, ma'am? Amino acids are there in the cytoplasm. How did they come, ma'am? Through nutrition. Through nutrition, through digestion. All these amino acids are there. So these amino acids are brought in the correct order. Child will ask, how does the tRNA molecule knows the order, ma'am? Yes, it knows. Because author is only giving the transcription for mRNA. He doesn't say the transcription for tRNA and rRNA. Please teach them when mRNA is getting transcribed, the required tRNA will also be transcribed. The required rRNA will also be transcribed, so that the tRNA molecules will know which amino acid to bring first, which amino acid to come next. So what I normally do, I use a ruler. In the ruler, there is a 5' end and a 3' end. This I'm using only a pen here. I use one either small chalk or the eraser. This eraser is the ribosome, or the chalk piece is the ribosome. This is the 5' end of the mRNA, 3' end. See, the ribosome moves codon after codon. I show them slowly move the eraser. As the ribosome moves, as the ribosome moves on the codon, the particular amino acid will be brought. How it is bringing? Because same time the DNA has also given. That is why DNA is the master control. DNA knows the sequence of amino acids to be present in the mature polynucleotide. DNA governs the process, dictates the process. Then the child will ask, then why, ma'am, it is not taking place inside the nucleus? Why DNA is not coming out? Since it cannot come out and govern the process, it is sending a messenger molecule. I normally give an example. Principal madam, our principal madam or sir will be in the principal's office soon. Principal wants to convey something to a 12th standard child. How principal will convey? He will send a circular. Who will carry the circular? The attender will carry the circular. Supporting staff. So the supporting staff does the job of mRNA. Principal gives the info. Principal is the DNA. Principal is the DNA because he wants the 12th standard children to. Principal is elsewhere. 12th standard class is elsewhere. DNA is elsewhere. Ribosome is elsewhere. How DNA can govern, how principal is having the control over the changes, actions in the class, because he sends a messenger. That messenger is carrying the circular. Circular is mRNA. Because, and we read the circular, we decode. As teachers, we decode. So, like this, if you give examples, children will understand translation. For some reasons, DNA cannot come out. If it comes out, it will get damaged. There are many hydrolytic enzymes, they're all enemies. DNA cannot be present in the cytoplasmic environment, so it can't come. So it has to govern the process, right? Remote control. So it is able to control it remotely. It is sending a molecule, messenger molecule. That messenger molecule comes to ribosome. Ribosome does 2 important jobs. It gives place. It also gives enzyme, ribozyme. So the entire catalysis takes place. Same, the tRNA is also transcribed. So the tRNA knows first amino acid to bring is methionine. So show them the movement of ribosome like this. Simple. By then, polypeptide will be formed. Polypeptide will be extending. That polypeptide will come out, termination process. There will be a release factor. Release factor will be at the end of the 3' end. This release factor will nascent polypeptide chain. This polypeptide chain, the length of the polypeptide chain depends on the length of the mRNA. There are short polypeptides, there are long polypeptides. Insulin has 3 polypeptide chains. The pro-insulin has 3 peptide chains. Like this, you need to give them some simple models and teach them. Then it becomes very simple for, and like replication, translation also teach them the machinery, raw materials, from where the raw materials came, how the raw materials are used, how the process goes on. The say one paragraph will be there in the reader. That paragraph will tell about initiation, elongation, termination. Make the children divide the paragraph into 3 portions. First portion is initiation. Second portion is elongation. Third portion is termination. What they learn is very simple, but what we do is we teach them in detail. Then let them go to the reader. When they listen to us in detail, then the reader information becomes very simple for them. But when we teach only from the reader, they become confused. As I told you, in so many places, author is not explicitly talking. He doesn't say methionine. He only says it's a dual codon. He doesn't say whether methionine will be there in all the polypeptides or not. Like this, author is hiding a lot of information. You need to do a lot of research as teachers. Keep ourselves updated. There are a number of books, biotechnology books, molecular biology books, websites, Wikipedia. Visit them. You make a lot of supplementary notes in your reader. Give them all these information. The lesson becomes less complex. Otherwise also, in spite of doing all these things, children will find molecular basis chapter little difficult at the end of the year. So you need to, during revision class, so take lot of time, teachers. Don't worry about the number of periods. You take number of classes. It's okay. There are some lessons where we can do a very simple, like microbes in human welfare can be very easily done. Ecosystem can be easily done. Whereas these chapters need more time. Take extra classes if needed. So I believe a lot in very strong teaching so that revision becomes easy. There are some schools where they finish in such a hurry. They will finish there. Don't worry about others completing syllabus. Don't worry. Suppose your counterpart is some other school. I finish molecular basis. You don't get perturbed. You take your own time. Take your own pace. Go in your own pace. Every day recall. Give them lot of hypothetical examples. Give an RNA, ask them for the DNA. Give the coding strand, ask them for the template strand. Give the mRNA, ask them for the template strand. Do it in a reverse way. Simple mathematical calculations. Calculating the percentage of adenine, uracil, etc. All these will make the chapter simple. Otherwise, the lesson is a nightmare. It is a nightmare for 12th standard children because they have not been exposed to such high levels of molecular biology till 11th standard.